And the last talk of our session this afternoon will be by Anna Neto, Dissecting the Mechanism of Oncogenic Glutamate Receptor Signaling in Melanocytes and Melanoma.
Hi, I would like to thank for the opportunity to present my work today. I'm a postdoc in Craig Seal's lab. So today, as Liz already said, I'm going to talk about dissecting the mechanism of oncogenic glutamate receptor signaling in melanocytes and melanoma. So, melanoma is the most deadly form of skin cancer. It kills approximately 10,000 people in the US annually. And these melanomas, they arise from melanocytes. So if you see this skin scheme, you can see that melanocytes, the pigment-producing cells, they are located in the lower part of the epidermis, and they produce, this pigment is called melanin. So when these melanocytes are altered, they start to spread deeper in the dermis until they are able to reach lymph nodes and vessels, and then they are able to spread throughout the body. Approximately 50% of melanomas, they harbor BRAF mutations, and 20% of melanomas present RAS mutations. But these mutations, though they are critical, but they are not sufficient for melanoma initiation. So there's a need to look for additional genes. And melanoma is a highly unstable genomically. And sequencing studies allow to identify many genetic changes in melanomas. Here in this image, you can see the representation of the sequencing of only one melanoma. And from the comparison between the tumor and the normal tissue, throughout the genome, they were able to identify the several DNA-based substitutions represented in orange, several copy number variations, and several rearrangements in green. But the question that needs to be addressed is which ones of these mutations are really relevant? But we focused our interest in one particular gene, and we used the model system to address its function. And which is the gene is the GRM3 that is recurrently mutated on several studies, around 15% of all the melanomas that were tested. But it remains to be clear if it's a bona fide gene and how it functions. And why is that? Because it has an unusual profile of mutation. So it appears heterozygous as an oncogene, but it shows several mutations throughout the gene as a tumor suppressor. As you can see in the protein structures, and the several mutations are represented throughout the protein. And one of the hotspots is the E870K that is represented in several of the melanomas that were sequenced. So what is the functional consequence of these mutations? So the first question is, what is GRM3? It's a metabotropic glutamate receptor 3, a G protein coupled receptor, and glutamate is a major excitatory neurotransmitter of the central nervous system. But the role in melanocytes is kind of unknown, and this glutamate receptor 3 is able to regulate several downstream pathways, but from AKT, cyclic AMP signaling, and MEK, we don't know which one is the most important. So we take advantage of our favorite model, Zebrafish, as you could imagine, that, as you know, is a valuable tool to study melanocyte biology. And why is that? It's very clear. As you all know, it has a rapid melanocyte development. It has several single-cell resolution assays are available, and several pigmentation mutants are also available. And one of these great assays that we have available is the Mini Cooper assay that allows us to make designer melanocytes. So we take advantage of the MITFA mutant that is an essential, MITFA is an essential gene for melanocyte formation. And we are able to, and we use a TOL2-based vector, Mini Cooper, that has a MITFA mini gene, and that we can couple our gene of interest and this injection is able to rescue the formation of melanocytes in a cell-autonomous way. So we are able to get embryos that then can be raised that are chimeric, and that all of these rescued melanocytes, they have our gene of interest. So using this Mini Cooper assay, we use as a control of our experiment EGFP. So when we inject EGFP, we were able to obtain we are able to obtain melanocytes with the pigmentation that we expect with a dendritic shape. The same happened when we inject the wild-type copy of the glutamate receptor 3. But when we inject the E870K mutation, what we observed is that the pigmentation of these melanocytes was very different. And we tested three more variants, three oncogenic variants of the gene, and we were able to observe similar altered pigmentation pattern as you can observe, and that we were able to quantify. So it seemed that these tumor-associated variants of glutamate receptor 3 were able to cause an altered pigmentation. All the four variants, what could indicate that they are acting in the same downstream pathway. So we were wondering, what is this E870K mutation affecting? Is it leading to a smaller melanocytes, or are we having an effect in the melanosome? So the melanosome is the organelle that contains the melanin, or the pigment. And in this case, we can see that it's focused, that is located in the center of the melanocyte, that you can compare with the normal melanocyte, where the pigment is spread throughout the melanocyte. So to address this question, what we did, it was to use a membrane marker, kils, that drives M-cherry, so we were able to check for the melanocyte area. So here you can see, when we inject the wild-type glutamate 3, that the area of the melanocyte and the melanosome is pretty much the same. But when we inject the E870K construction with the kils marker, we could see that the area of the melanocyte is much bigger than the melanosome. So it means that it seems that the melanosome is aggregated in the center of the cell. And we could see that the ratio of the area of the pigment and the area of the melanocyte, it's much lower in the E870K derived melanocytes than when we inject the wild-type glutamate receptor 3. So it seems that we are observing that melanosome dispersion is affected in mutant glutamate receptor 3 expressing melanocytes. So it's known that cyclic AMP levels can regulate melanosome trafficking in zebrafish. High levels lead to melanosome dispersal, and while low levels of cyclic AMP lead to melanosome aggregation. So we were wondering if our E870K mutation was a consequence of the cyclic AMP levels. So to test this question, we used forskolin. That is a drug that activates adenylyl cyclase and increases cyclic AMP levels. So when we add the forskolin to our E870K derived melanocytes after treatment, we could see that there was a melanosome dispersion, so the pigment was spread throughout the melanocyte. But when we used an analog of forskolin that acts in the main different pathways that forskolin does, except adenylyl cyclase, so it is not able to increase cyclic AMP levels, we were not able to see a rescue of the melanosome aggregation. And when we use caffeine that is able to indirectly increase cyclic AMP levels, we were able to see a melanosome dispersion showing that increased cyclic AMP is able to suppress melanosome aggregation caused by the E870K mutation. So our results so far show that the E870K mutation is upstream of adenylyl cyclase, being able to inhibit the decreasing cyclic AMP levels, what leads to melanosome aggregation. So our next question is if this mutation is able to also lead to the reduction of cyclic AMP levels in melanoma cells. So what we did it was to express in the A375 melanoma cells the wild-type glutamate receptor 3 and the E870K version of this receptor. And we were able to assess the cyclic AMP levels. And when we used the DCG, an agonist of this glutamate receptor, we didn't see any difference of the cyclic AMP levels when we compared the wild-type receptor with and without the agonist treatment. But we saw a significant reduction of cyclic AMP levels in the case of the E870K in the E870K with the drug treatment. So it seems that the E870K mutation causes an agonist-dependent reduction of cyclic AMP levels. So our results show that tumor-associated glutamate variants cause altered pigmentation, and the disruption of melanosome trafficking by oncogenic variants of glutamate receptor 3 results from altered cyclic AMP levels, and that the E870K mutation also lowers cyclic AMP levels in human melanoma cells. And these low cyclic AMP levels is related with melanoma initiation. It is known that high pheomelanin leads to melanoma initiation, and these low cyclic AMP levels are also related with progression with high proliferation on metastatic cell lines. I would like to acknowledge my lab, especially Craig is being a great supporter, and our funding resources, especially my NRSA postdoctoral fellowship. Thank you for listening.
These melanomas, produce metastasis?
In the mice, the A375 that express the E870K, in the mice they are able to produce metastases.
But how about your model?
I didn't do it in fish.
Okay, thank you.
I didn't test it in the fish.
I had a question. Maybe, so these are driven from the MITF promoter, and so then we're seeing in the differentiated melanocyte effects. But have you tried driving it earlier to see if it has an effect on the development or the proliferation of the melanocyte lineage?
No,
to get an idea about
tested.
Initiation and...
You were saying that to try to use another promoter to express even earlier.
Yeah.
We haven't tried, but could be very interesting.
Okay, thank you very much. Okay, so that's the end of the cancer session, and I would just like to thank all the speakers one more time before we finish. So thank you.